Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Stephan Ripke will present Genomics across diagnostic boundaries to improve precision medicine in psychiatry. The Brain and Behavior Research Foundation is committed to alleviating the suffering caused by mental illness by awarding grants that will lead to advances and breakthroughs in scientific research. The Foundation is the largest private funder of mental health research grants. Since 1987, the Foundation has awarded more than $380 million to fund more than 5,500 grants to more than 4,500 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists who are working to find breakthroughs in disorders such as addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. I'm delighted to introduce Dr. Stephan Ripke. Dr. Ripke is a university professor in the Department of Psychiatry and Psychotherapy at the Charité campus in Berlin, Germany, and a research scientist at the Analytic and Translational Genetics Unit of Massachusetts General Hospital's Simches Research Center. Dr. Ripke was a 2015 Foundation Young Investigator grantee and was awarded the 2014 Sidney R. Bear Jr. Prize for Innovative and Promising Schizophrenia Research. Today's webinar will begin with Dr. Ripke's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit your questions throughout the presentation. Following the presentation, I'll present your questions to Dr. Ripke and will address as many of them as possible in the time allotted. And now I'm pleased to introduce Dr. Ripke. Stephan, the floor is yours.
So, do we want to switch on the webcam, Nico?
Yes.
It should be on, and if you can accept to share your desktop, please.
Perfect.
Thank you. Perfect. Good. Thank you. And the video cam, please. Okay. Perfect. Now we need to swap back to your presentation. Perfect. Good. Thank you.
Perfect. Wonderful. So thanks, Jeff, for this very kind introduction here. So as already mentioned, my name is Stephan Ripke. Good morning, good afternoon, good evening, everybody. I'm not used to webinars worldwide like these, even though I'm, of course, used to many phone calls internationally, so at all different time zones, even though I understand that most of the audience here will tune in from the United States, as far as I understand. Today I want to give you a very personal 1-hour introduction to the world of psychiatric genetics, and especially into this one successful analysis method of the last decade, Genome Wide Association studies. And I understand that many people here in this audience are actually lay persons, and I hope I will not become too technical for you who are not dealing with these kind of analyses on every day there. So, but let's jump just right in. And I hope this is not proceeding. So we tested this before. So Nico, you don't see the next slide, right?
Not yet. Can you click one more time on the slide, please?
Nope.
Okay. All right. Try with the arrow at the bottom left corner. Drag the no. Okay.
So now it's working. That's fine.
Yep. Good.
I think before you switched on the slides and it didn't work. So when I switched it on, it's fine. Good. So okay. let's jump right in. So this slide actually should illustrate everybody that these tools or devices that we do have in classic medical diagnoses, and especially in classic medical research, for almost all of these diseases, we have one of these devices and tools that you see here on these slides, like be it even like complicated things like MRI scans or even animal models in here, or if you just measure the blood pressure, which obviously is but these objective devices that give us some numbers in here, or something that we can really look into detail, be it also a microscope or like biochemistry in here. And what I mean here with these classic medical diagnoses, we are here with the Brain and Behavior Research Foundation, obviously dealing with a disease that is somehow different to these classic medical traits, let's say it like this, and this is psychiatry. And especially here, if you look at this one disease of psychiatry, but it's actually true for many, for almost all of psychiatric disorders in here, that we're dealing with a disease in here that have a wide range of symptoms in here, or also cognitive dysfunctions, etc. and so on. I don't want to go into detail here. I'm not a psychiatrist, and psychiatrists are doing a much better job in presenting this disease by itself, but what is very clear here is that in psychiatry we are missing these devices, these machines who actually help us in diagnoses and actually also help us in research here. So the tools that we have in this medical area, as we all know, are our eyes and our, like, the psychiatrist who has to observe and to speak to the patient in order to get to a diagnosis. Without a patient, there's definitely no diagnosis to be made here. And on the same second, and that's the interesting part here, on the same second we are treating, well, different psychiatric disorders, of course, differently, but schizophrenia is a good example that we're actually treating this disease predominantly pharmacologically. So on the biological side, even though we don't have clear biological hints for that, we're treating that biologically. And this is the drug chlorpromazine that at least the psychiatrists of you have definitely dealt with multiple times. This is the first drug that came out for treating schizophrenia here. The drug characteristic blocking the DRD2 receptor is still the central mechanism when we treat psychotic diseases in here. So this is to keep in mind, DRD2 receptor blocking, very important. And what we know about this drug is that it has like a wide range of side effects. There are common side effects that are not as severe, but they occur, as they say, commonly when you administer this drug. But they have also serious side effects that obviously are not as common there, but of course problematic for giving this drug. But even though we have this wide range of side effects, the World Health Organization is still listing this drug as one of these essential medicine, and quotes one of the great advances in the history of psychiatry. So, and if you read the reports, what happened when this drug was first introduced in the world of psychiatry, then you understand what the big difference now was. Before, you had basically nothing in your hand than maybe the prisons and to lock people up who are not doing well, and suddenly you have a drug that gives at least a portion of these patients their life back and their possibility to work and to enjoy life back. And so what is special about this drug, and this is something that we all have to think of all the time, is that this drug has been found in 1950, and especially not in psychiatric research, but in anesthetic research. So this is a purely incidental finding. And all of currently used drugs for treating schizophrenia have still this drug mechanism at least in part of the blocking the DRD2 receptor. So actually we didn't, since the 50s, 60s, we didn't introduce a profoundly new drug mechanism that can have a similar effect as this drug discovered many decades ago. So that's exactly what Steve Hyman called here in 2012 the revolution stalled, and he's listing not only chlorpromazine, that's what you see here in the middle, but he lists also all the other drugs that are commonly used in psychiatric treatment, be it lithium as a mood stabilizer and the antidepressant. All of these drugs, common to those is that they were discovered in the 60s or 50s, and since then we didn't find many new mechanisms in here. This plot actually, unfortunately, I forgot where I found this plot, but this illustrates it again very nicely how psychiatry lost the race against like internal medicine since the 50s, for example, where like for heart disease we got really a huge number of new mechanisms in here, and schizophrenia and depression therapy is almost unchanged since the 50s in here. But there's one thing that like psychiatry or psychiatric diseases have in common to classic medical diseases from other fields in here. And this is here the heritability. So something is hidden in the genes. And this is something that we know from twin studies. For this kind of analysis, you don't need genetics in that very sense. You actually only need epidemiology with observing twins, comparing monozygotic twins with dizygotic twins, or monozygotic twins in the same neighbor environment with monozygotic twins that are raised in different environments, etc. So analysis, we know the prevalence of a disease. So these are all diseases that have at least 1 in 1,000, if not 1 in 100 or 1 in 10 prevalence. So fairly common diseases in here. The abbreviations that you see here are like type 2 diabetes, Crohn's disease, breast cancer, so classic somatic diseases in here, where we know a lot of biology already, and they share with these complex, common psychiatric disorders, schizophrenia, bipolar, or the much more common disorder, major depression, they share a fair amount of heritability. That's what you see on the y-axis. So there is definitely something in the genes that's inherited from one generation to the others. And that's where genetics kicks in. That's where genetics will hopefully lead us new ways in trying to do some research with these devastating diseases in here. So maybe a short recap on genetics, and I think this might be worth repeating here, that we have basically two big arms in genetics. So there's this first arm, and I will give you a couple of details in the next couple of slides here. The first arm is really these classic Mendelian disorders that you think of if you think about a genetic disease here. Again, a couple of examples will follow here, where we actually also not only want to find out something about the disease, but really about the individual. We want to predict something about the individual based on its genetic configuration. But the second part is sometimes a little bit forgotten, and this is the most, in my opinion, the even more important point here, that via genetics, we try to understand the biology of a disease, and in order to design better treatments, not directly for the individual, but for the group of patients with these diseases in here. Let me give you a couple of examples from these monogenetic traits, let's say. For example, a very easy monogenetic trait is here, the blue eyes, for example. If you have two parents with this recessive trait, blue eyes, the likelihood is very high that the offsprings will have blue eyes as well. This is the classic recessive inheritance pattern that you see with a trait that you can observe everywhere. Of course, I want to mention this is nature has its own rule, and there are exceptions to these rules. So these are really just examples to make the point, and will not always be true here. Another trait that's exactly on the opposite, because it's not like a nice healthy trait, but it's actually a very severe disease, Huntington's disease. Probably many people have heard about it. It is a disease that has some overlap with psychiatry. So we have here mood and mental problems in here, especially in the beginning, but even in the end, it will decline into dementia in here. But there's a lot of neurologic coordination and movement problems combined with that, and actually with our, that's what I mentioned before, with these devices like an MRI, we can actually even help this diagnosis a lot. But to be very clear, this is a disease that is diagnosed via genetic testing in here. As if you see this list here, a couple of epidemiologic traits that I put in here. First of all, it's a pretty rare disease. It's definitely not in the range of schizophrenia, where we have around 1,000 people out of 100,000, but it's just only 4 to 15 in here. It's definitely inherited almost always, not always, but almost always, that if somebody has the disease, it got the disease from one person in here. This is hidden in this autosomal dominant word in here. But again, the diagnosis is done via genetic testing. So we have here this classic term like personalized medicine or whatever. So it's really like you do the genetic test and you know about the disease almost certainly. Even though we don't know exactly when the disease will occur, we will be able to tell that the disease will most likely come to this person. Even though, and I want to also mention that even though we know about this gene since a fairly long time, we didn't find a cure for Huntington's disease. So finding a gene doesn't necessarily mean we find the treatment. But I will give you a couple of examples later that there are also many examples where it actually does help for treatment. For example, this is a very beautiful example here, where we have also a Mendelian trait here, PKU. Almost everybody who has a child in the US has heard about this disease, because what actually happens, again, it's a very rare disease, but what happens, there's a very strong newborn screening carried on in the US for this disease, because interestingly enough, and again, this is just, it is basically a genetic test that is done here. So because interestingly, if you're not reacting to if you're not screening, then this relatively rare occurrence will have, with just normal diet, will have severe intellectual disability and other serious health problems. But if you know about this genetic defect, you can maintain a very strict diet and have a very normal health and normal lifespan. So treatment, even though it's not even pharmacologic treatment, can have a huge effect here on this genetic test on this monogenetic disease. So huge success here in this point here, finding also out about the biology of this disease in here. So what you can see here in pink is this, and here you have the years going by starting from the 80s, but especially starting in the 90s, where we actually did find, for these Mendelian traits that I actually put out these examples right now, where we could find genes, usually one gene for one disease in here, via this analysis method called linkage analysis. So you see this huge success that we had before the 2000s for these, and this success is still continuing. We find genes for these rare monogenetic diseases in here. But what we see here also is this blue line that's actually almost not moving here till the year 2000. Mark Daly called this the dark ages of complex trait genetics in here. So for these so-called complex traits, and I already introduced these complex traits meaning like many different influences coming in here, also environmental influences, but that's what we know already now, also many different genetic genetic contribution from different points of the genome. So not only one gene being a part in this game here, but also many different genes in here. These are these so-called complex traits in here. And these complex traits, we didn't find out much of that, even though we knew they were heritable. The so-called linkage analysis didn't help us much more in here, because these variants that are connected or associated with these diseases do harbor only relatively small effect sizes. So they're not deterministic for the disease as these monogenetic diseases are. And what comes now into play is these genome-wide association studies, GWAS. This is currently the golden standard for these complex disorder analyses. And what you do here, you do not collect families as you did for these monogenetic diseases, but you collect cases and controls, and you test them against each other. And what is important here to understand is that what we are testing here, we're not trying to find new genetic variants that actually that will determine the disease, but actually variants that are already found in the healthy population and just change their frequency in the cases versus the controls. I will go into that in the next two slides in a little bit more detail, but for those of you who are not very familiar with these kind of analysis, just as a summary here, that we're actually testing almost up to 7 million, sometimes even more, sometimes a few less, of these variants that are standing variants in the normal population. We test the whole genome in here. And since we are testing so many different points in the genome, we need a very strong evidence. And evidence, I use evidence here as a word for significance. We need a very strong significance in order to call a variant being unambiguously associated to a disease. And in order to get very strong evidence, most of the time we need big sample size. And this is here the take-home message here, the big sample size for these analysis method, genome-wide association study. What you see in this lower part of this plot in here is this so-called Manhattan plot. I must admit, I don't know which disease this is now coming from, but I want to explain you this Manhattan plot because you will see a couple more in the next couple of slides. You see here the chromosomes 1 to 22 on the x-axis, and these are actually also on the chromosomes. They are the positions also depicted. You see a lot of dots in here, so many dots that actually in the lower part of this plot, they're even merging in here, but these are actually just 7 million dots that are plotted there. What you see on the y-axis is the so-called minus logarithm of your significance. Meaning, for those of you here who are familiar with significance, we usually require significance values of around 5%, meaning this is a number of 0.05. Since we're testing so many variants, that's what I already mentioned here, we need strong evidence, so we are not requiring 0.05 anymore. We are requiring 5 times 10 to minus 8. This is a 5 with 7 zeros before that and then the comma before that. So that's why, since it's very difficult to plot this, we actually take the minus log of these values in here, and this is what you see on the y-axis. So meaning a 5 here means a 10 to minus 5, and this line here is this so-called 5 times 10 to minus 8, what I just mentioned in here. This is our genome-wide significant threshold. And so what we're looking for in these Manhattan plots are these skyscrapers that you see here. So these skyscrapers actually indicate they are passing our genome-wide significance threshold. This is the sky here of this dotted line. They're clearly passing way above this line, and you see so values up to 10 to minus 16, etc., and so on. And so these are the regions that we would call genome-wide significant. And for this specific disease in here, again, I don't know the disease, it would be 1, 2, 3, 4, 5 regions that we would claim of being probably unambiguously associated to the disease. Most of the time, especially in older times, you still needed to replicate those findings, but these are, from a first look, very stable results in here. Just to depict it. So how does this look like? Coming back to the single variant that we're testing, and I think that's important to understand. So these are let's call it first just an association study, not a genome-wide association study. This is just for one of these variants. And as I told you before, these are variants, and maybe coming back, this is a word that you want to keep here, single-nucleotide polymorphisms, a SNP, so-called SNP. So this is one of these SNPs. And usually in these SNPs, you carry two different alleles. Every individual carries two different alleles in here. And so what is shown here is that, like in the controls, we have actually a couple of controls who carry allele 1 and not allele 2 in here. So these are these red individuals. They are perfectly healthy, and they carry this allele, which might be associated to a disease. And from this test here, we actually know, just from the cartoon, it is associated to this disease. Many cases, actually, many more cases carry this allele than the controls do. And this is basically the test that we are doing in here. We're just doing this test, as I said already, more than 7 million times. And that's why we need very big sample size for the strong evidence or strong significance, what I just mentioned. So how do we achieve this big sample size? So there are a couple of technical, technical things that you clearly need for in order to achieve big sample sizes. You need technology that is affordable. These are these genomic chips where you just put a blood dot or like a DNA on there, and you get actually measured these millions or hundreds of thousands of SNPs in here. You need genomic resources. Now I will come back to that in a second, why you need that, but you probably have heard about the Human Genome Project, and the Human Genome Project was in the beginning of the 2000s, so that's, you might now understand why this kind of analysis came actually later. But the number one thing that, in my opinion, that you need for big sample sizes is collaboration, because not a single university can collect these numbers that are mostly needed for most of these analyses that we're doing. I want to do some two technical courses about what is genome-wide association studies, like some two central technical terms in here. There's this technical term called imputation, where we're actually using so-called linkage, this equilibrium, in the reference data set. And here comes into place what I just mentioned, the genomic resources that we need, formerly the 1,000 genomes resource. Meanwhile, the haplotype reference consortium is the classic up-to-date reference that we're using. And with this imputation method, we are now actually not, we are increasing the SNPs that we're measuring from these, be it 300 to 700,000 that we are measuring on the SNPs, on the chips, and we are increasing this to many million variants, and can apply and associate, analyze these million of variants directly, even between different data sets. That was a very important step to actually bring data sets together to do this. Another technical detail about genome-wide association studies is that even if you analyze ancestry specific, so meaning even if you analyze data sets that are collected with European ancestry, you see differences between the individuals. And I want to show this via this plot here that is actual data from our consortium in here. And what you see here is that these individuals that we are testing here, all of these individuals, probans, schizophrenia cases, and controls, they have a dot here on this plot. So it's like around 70,000 individuals that are plotted here, and we are using the genetic information to distribute these individuals over the scatter plot in this piece, so-called principal component 1 and principal component 2. So we are extracting the main information from this genetic differences, so we can distribute them over this plot based on the genetic information, and I colored coded them based on the country where they are from. And what you can see here very interestingly is that, for example, the Finnish data set that is here is a strong outlier to the if you want to turn in your head a little bit this slide, that this is the strong outlier in the northeast in here, like merging into these other Scandinavian countries. These are the Swedish and the Norwegian countries. This is then continuing into these middle European countries, especially Germany in the center here, Bulgaria and Portugal comes more in the south, and then in the way south in here is this Ashkenazi Jewish component from our US-based samples in here. And basically most US-based samples are scattered in this triangle in here. So interesting, not only an interesting concept in here, but also something that we have to take care of while we are doing these analyses. So how did these genome-wide association studies develop in the time scale that I just showed you before? So we have here, and again now the time scale starting from 2000, and you see here starting from 2006 this explosion of findings from just one disease in here. And this is now this different concept that we have all to understand. So this is just one disease, Crohn's disease, and we have 122 genome-wide significant regions, probably pointing to many more different genes in here that seem to be, or that are for sure associated to this disease in here. So it's not only one gene that has something to do with the disease, but it's really many, many genes. So this is this polygenic disease that probably many of you have heard about. And what you can see on this plot here as well is that we do not only have this explosion of findings, but we actually also have this explosion of co-authors in here that you see here, and just reflecting this nature of collaboration that I mentioned before. Really many people have to come together to bring together a sample size that is sufficient for these kind of analyses in here. But not only Crohn's disease, but many disorders were very successful with this kind of analysis method. With these GWAS, genome-wide association studies in here, what you can see on this plot here is also a classic plot that has been shown before many times. You see the 22 chromosomes here, chromosome 1, 2, according to their size, and you see with all these colored dots, all these diseases that have been mapped to some parts of the chromosome, and you can see that almost all chromosomes have been mapped multiple times with a huge plethora of different disorders in here, including psychiatric disorders, and we will get back to this in a second. So did this actually help us in treating patients, giving us the possibility to actually help a patient for their treatment or so? And I cannot give you very good examples for that, because almost all of these analyses are still pretty recent. But what I can give you is a short list of hindsight examples where we actually do find the genes that are currently used drug targets with these kind of analyses. So these analyses clearly have the power to find the targets for our drugs that we're currently using. Obviously, these drugs that you see in this list here have not been found with genome-wide association studies, but they could have been if we didn't know about them before. So this was a very long introduction to genome-wide association studies. Now we are switching to genome-wide association studies in psychiatry. And if you understood the success in other medical fields, then you could probably understand why the hopes were so high in this disorder. And this here is the slide from 2009 that got everybody a little bit disappointed back then. It is already a consortium. It's the International Schizophrenia Consortium. And you see this also based on the numbers. So there are already 2,600 cases collected. This is a big number to be collected, usually not possible from one center alone. And in this case, it was a couple of centers together. And when you see this Manhattan plot that I actually described to you before, then you see that none of these skyscrapers actually pass this genome-wide significance level with the red line, even though these are numbers that where Crohn's disease was already utterly successful in these sample size. So you have 0 genome-wide significant sites, and disappointment was big, but the field continued to collect and to analyze data sets with schizophrenia cases and controls, and a new big consortium has been built, and this is exactly this central consortium where I am part of, but definitely not me alone. There are almost up to 1,000 members. Meanwhile, we have many different institutions from many different countries. They're really coming together to build this consortium in here. And there are two special things about this consortium, actually three, but the third one I will go back later on. But so two special things here. First of all, we are collecting raw genomic data from many different individuals from a plethora and different psychiatric disorders. So not only schizophrenia, but also bipolar, major depression, and many more diseases in here. And what you can see here is our biggest success story so far from 2014 Nature publication, where we actually could publish an analysis that consisted out of 35,000 cases and 46,000 controls. So you see here this huge increase of sample size that was done within only 5 years in here, and this sample size was then again sufficient enough to show us really almost 100, and with replication more than 100 genomic regions that are clearly associated to this disease. So a huge success story, only possible with really many people strongly working together to bring this beautiful data set together. So did this help us did this give us new insights? First of all, we could confirm some old insights or hypotheses that we have in schizophrenia. For example, the glutamatergic hypothesis. These are plots that are very similar to the Manhattan plot, just zooming into single genes in here, and we could here, for example, confirm or at least give more support to the glutamatergic hypothesis. But I think most importantly, and I think this loops back to one of my first slides in here, is that we did find this DRD2 gene, this DRD2 receptor, being genome-wide significant, associated again with this kind of analysis. So we actually also here, we find in hindsight our drug target, our number drug target in schizophrenia therapy, giving us big hope that one of the other 100 regions that we have in here will also harbor one of these drug targets that from the future. That's exactly what we're hoping. One of these examples, for example, the calcium channel, many different subunits throughout over the genome, and for schizophrenia we have actually 3 different subunits back in 2014. Meanwhile, we have even more here that are actually associated with schizophrenia. So of course this is a gene that is expressed in the brain, and it's like under big scrutiny if we can use this therapeutically for treating schizophrenia. A small excursus now into bipolar, because actually this calcium channel that I just mentioned has been found before in bipolar, even before schizophrenia, just to mention that. But this actually gives us already this interesting phenomenon that we see and observe all over again, that we do see some commonalities between the genetics of schizophrenia and genetics of bipolar, and I will get back to this in 1-2 slides as well. So if we look at the history of schizophrenia, GWAS, and you see here in the green line, you see here what happened here, the number of findings in this gene on the y-axis and the number of cases on the x-axis. And you see here, after we passed a very big sample size of around 10,000 to 15,000 cases, we are almost on a straight line where we do find 4 new regions for every 1,000 cases that we add to our collection. And you might ask yourself, does this just continue? Do we just have to increase our sample size, and we do find even more? And indeed, that's exactly the case. That was 2014, and we have now 2018, and we have here, this is the dot that we just looked at beforehand. We now have again doubled our sample size, and we are still on this very straight slide in here up into the future. So this is the Manhattan plot that we are currently looking at, just presented at the World Congress of Psychiatric Genetics in Orlando last year with 65,000 cases. Again, here doubling our sample size and almost tripling our number of findings that we have in here. So it looks like the whole genome is now associated. That's indeed not the case. These dots are just way bigger than they are actually on the genome, of course. It's still only a fraction of the genome, but it's definitely like it is definitely spread over the whole genome. Every single chromosome has some variants that are associated with this disease. Interestingly, so you might imagine that if we have so many genes, for some of these analyses, it will get even more complicated. And does this actually just add more frustration because we have to look at so many genes in parallel in here? No, but we have actually a very strong bioinformatics unit also at the PGC, Daniel Postuma, with this beautiful tool where we actually can have bioinformatic information directly integrated into these results very quickly and work with these kind of results in a very quick way. And interestingly enough, the more hits we have, the clearer the results actually get. So it's not introducing chaos, but actually introducing clarity with these bigger numbers of findings that we have in here. I want to switch topics a little bit, coming back to 2009 here. The so-called polygenic risk scores. Everybody who is working with psychiatric genetics has heard about them, but maybe for the laypersons, it basically means back in the day in 2009, where we didn't have any genome-wide significant findings, it made the point that we do have commonalities between two different samples for schizophrenia from two different countries. If we try to see, is there some commonalities between these data sets? And indeed, Sean Purcell back then could prove that this is the case with high significance in here, that we see some commonalities in here. Meanwhile, we know that this so-called coheritability between these two data sets is almost one. So there's a very strong same genetic architecture between two different data sets in here. What is interesting in here in this plot, that's why I want to show it in here, in the blue box, you see the commonalities basically between schizophrenia and bipolar. And you see here that this power of these polygenic risk scores from schizophrenia into bipolar is significantly reduced to schizophrenia, but it is still there, especially compared to other disorders like type 1 diabetes, etc. So it's still there. And this again makes the point of like this commonalities between schizophrenia and bipolar, which we observe very frequently and repeatedly in here. There's one concept of these polygenic risk scores that I want to touch here, is that the power of polygenic risk scores do increase with increased sample size. And that's what you see here. This is the same plot that you just saw from 2009 with our sample size of around like 3,000 cases in here. And this is our power of the polygenic risk scores, up to 20% explained variance if we use our sample size of 35,000 cases in here. So clearly this improvement of our polygenic risk score power in here. I want to quickly go into more detail about these risk scores because they touch this topic about genetic testing and disease prediction in here. And what you see here is the histogram of the controls in red and the cases in blue, and on the x-axis actually the normalized polygenic risk scores. And what you can see here is that these two curves of the cases and the controls are distinct from each other, but not completely. They're not completely separated. So if they were completely separate, this would be a very nice diagnostic test. In this case, you can state like even if the person has a pretty high like more than one standard deviation high polygenic risk score compared to the mean, it is the likelihood that it is the case is a little bit more than the controls, obviously, but it's still highly likely that it's still even also a control is possible. And this is the same also for the low scores. There are many cases also who have the low score. So this is not, as I say, yet, it's definitely not yet a diagnostic test, the polygenic risk scores, but they are used very often in research. Family history and severity are actually touched by that a lot. So I want to switch now to a very local collection in here that we're doing here in Berlin, especially because the BBRF funded this project in here. This is the Berlin psychosis study, where we actually try to, with I would not say minimal funding, but of course this is not funding for a cohort that takes millions of dollars, but a little bit more reduced funding in here. But we could actually, with this funding from the BBRF, relatively quickly collect and build a cohort of relatively moderate size. Let's say it like this. It's definitely not, doesn't belong to the bigger collections in here, but also not to the much smaller collections in here, within a time period that was relatively quick in here. And the most important thing is here that almost all of these individuals that we are collecting here are recontactable. So we can actually come back to them if we find possibilities for genetic testing, for therapy, etc., and so on. And this is the most important part here. And this is what I want to present here, because this is how I think nowadays you have to build new cohorts. Make sure that you can contact the individuals that you have in here again, because this will be interesting for the future, given that the polygenic risk scores will improve even more. So our polygenic risk scores for our BEPS Berlin study are pretty much in line with all the polygenic risk scores that we usually see. So we see this nice distinction of these two curves from the controls in black here and the cases in red. But again, not completely separate in here. That's fully in line with our expectation. We are proud that we actually, this relatively efficient cohort building that we are copying this right now in different cities in Germany, but also for example, in Vietnam, we have a nice collaboration where they collected within an incredibly short time, just a couple of months, they collected a pilot study of 200 schizophrenia cases and healthy controls. We currently genotype them, and if we are happy with the quality, etc., then this will definitely a project that will be continued. And speaking of Asian contribution, I want to give a shout out to the Asian schizophrenia group in the PGC, that we're able to collect already also a very big sample size in a relatively short time frame, though they collected also some data sets that were out there since a very long time. But we have many more that are collecting here right now, building cohorts. So interesting that we are also increasing our diversity for ancestry in here, and that was very important and gives us a lot of insights in here. I want to switch a little bit. Diseases. This is a Manhattan plot from bipolar disorder, currently in the bioarchive, but hopefully to be published soon. Very similar genetic architecture as schizophrenia, as I mentioned before. And also we get around 30 loci with 20,000 cases, so it's a little bit fewer than with schizophrenia, but in the same ballpark, let's say like this. And this is different to this study that has been published, I think 1 week ago, where we actually did find 44 risk variants for major depression, where major depression for a long time, many people thought this will never work, that we do find some variants in here. it happened. So we actually could find this number of variants in here, but it was necessary, and that's what you see in here. It is a different genetic architecture, definitely. It was necessary to collect a much bigger sample size as we were used to in schizophrenia. I want to quickly scan through a little bit of secondary analyses that are in this manuscript in here from major depression. We could confirm, obviously, that we are not finding some random results in here. This is really brain specific, and not even brain specific, but neuron specific compared to oligodendrocytes and astrocytes in here. And this plot here very quickly should give you the overview that the polygenic risk scores that we are creating for major depression in here are very much similar behaving as the schizophrenia scores, just not as strong, but we have similar, leave one out, polygenic risk scores. We have similar behavior, that the higher your major depression score is, the more likely you have to get this disease in here. And interestingly enough, also these polygenic risk scores again work for these endophenotypes. Polygenic risk scores for major depression are associated to age of onset or to the severity of the disease or single episodes versus recurrent disease. So this is a very similar observation as we did for schizophrenia, only that we needed much higher sample size in here. Where we want to get to in the end is something like this, and I borrowed this now from a very different disease here, prostate cancer. Prostate cancer, obviously a complex trait in here, environmental factors in there, but also lots of genetics in here. But there is a test in here, and if you want to learn more about this, then you just have to remember this STHLM3, which stands for Stockholm 3 test. This is the Stockholm 3 test that actually combines not only like blood markers in here, but also genetic, they call it genetic traces, but these are basically SNPs in here, and some clinical data. They combine this and they claim, and they make a strong argument that they reduce the number of prostate biopsies. So this is now this first step into genetic testing from these complex disorders in here, and highly promising, and that's exactly where we want to get to if we speak about psychiatry, but we are clearly not there yet, I would say. As one of the last slides, I want to show here also one thing that I think the PGC, the Psychiatric Genomics Consortium, can be very proud of. Very early on, the PGC always decided on sharing their results openly. These summary statistics, so basically these meta-analyses that we are publishing, these Manhattan plots, and the actual data below that is shared freely on the web, so you can actually download them without being a member of any university. You don't need to apply for that. You can directly download them. We made sure that there's no chance to trace the individuals for those, of course, and for everything that needs more security. For example, the real genomic data, we actually want to share this as freely as possible as well, but of course we cannot, because we want to secure the privacy of our probants, of course, and that's why we set up a big system where we provide access to these raw genotypes to all our PGC members. And so, meaning the system is, if you become a PGC member, you're invited to work with the data to make more sense out of that, so that the science gets improved much in a rapid way. So this is the concept, and the idea of the PGC is to really share our results, and especially also our data in there. So I think, in summary, I hope I could make clear that I think these common variant analyses, these Genome Wide Association studies, were utterly successful in the last 10 years, that we need to continue to increase our sample size. And I hope that, like as we did here in Berlin, but many other smaller centers also can build these cohorts with with moderate funding in there, and I hope also the BBRF will continue to fund these kind of smaller cohorts here as well. And of course, this is clear that this is just the starting point. This is starting where we actually need to start working with the data and with our insights. This is not the solution yet. So far, we couldn't help treatment for anybody yet, and this is definitely our last goal. That before that we will not stand still, of course. I want to point to this small YouTube video from Ben Neal in here. In 4 minutes, he's actually summarizing what I did in the last 50 minutes to you guys, so a very beautiful 4-minute summary of that. And as the acknowledgement slides comes, of course, this is too short by definition. As always, there's so many people that I have to thank, and I will not get into details in here. Clinicians that are not on the slide who actually did the recruitment, I have to be very thankful. But most of all, I want to give out a big shout out and a very big thanks to all the people who gave us the genomic data, to actually gave us the trust to work with your personal data, to try to make sense out of that, and hopefully in the future we will be able to pay back with some improved treatment for people with psychiatric disorders. Thank you very much.
Stephan, thank you for an excellent presentation and for the work that you're doing. And I want to emphasize how this is a tremendous effort to have this level of collaboration among scientists around the world and people around the world really working together because you need these numbers. One question I want to ask you in our remaining moments. Obviously, as you said, the goal is to develop new treatments as a result of this. And the idea is that these Manhattan, where you see the skyscraper, might point us to potential treatments. How long do you think it's going to take for us to get there? What do you see as a time frame for that?
That's a good question, of course. I mean, and if I knew the exact answer, I would be a wizard, of course. So I would love to learn from other disorders here as well, right? So, I mean, Crohn's disease was so much earlier with their success in Genome Wide Association studies than psychiatry, and still I know of new treatments that are actually in trials for Crohn's disease. There are other examples for diabetes, where actually Genome Wide Association studies pointed to some drug targets that actually already are in trials. So there's a time frame of 10 years or so. But to be honest, for psychiatry, I think it will take even more than that. So my biggest hope is actually for a short time solution. My biggest hope is that, like, what pharma companies are already doing is that they take these hits, these skyscrapers that you see here, and they try to find in their compounds that they have in their cupboards, and to find compounds that are actually acting on one of the or two of these genes in there, and to find out if these are safe drugs in there, and to revisit them if they are safe drugs, to revisit them and maybe get into trials. I haven't heard that they actually did go into trials yet, but I have heard that they're actually doing this research about, like, looking into what do they have, what is safe to use. That's what I know that they're doing, and there's a big hope of myself. The other things, like the more distant solution, that you actually look at all these genes in here, you find one gene that is interesting, and you even go via animal models or like iPS cells, etc., and so on, would probably a time frame that's more in the range of 15 to 20 years or so. But that's a very personal opinion, and we all know sometimes it goes super quick because there's this one compound that we didn't think of before, and we try it out and it works. So I think there always needs to be also still some luck in there, but I think we are now on the path where we have something in our hand. So we have some light spots in this dark area where we can actually look for, and maybe we can force our luck a little bit more. But I don't want to put a specific time frame to that.
Well, I think your words about forcing our luck are great words, because in the past, as you said at the beginning, there might have been an agent that we sort of notice, hey, look at this, it actually does something else, and that may be helpful. And there's a number of drugs we could talk about in a variety of medical psychiatric conditions that fit that. And now we could sort of force that luck, as you say, because we have more information with which to test these medicines. This work is very exciting. Obviously, it will continue. I want to again say thank you for all that you're doing, because it will ultimately make a big difference in people's lives. I also want to thank everybody who joined us today. As you know, all of the research we fund is made possible through private donations. So please consider making a contribution by visiting our website, bbrfoundation.org, or call us at 800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or friend, please visit the events and webinars page on our website. And I hope you'll join us again next month when Dr. John Crystal, chair of the Department of Psychiatry at Yale, will present ketamine. Why now? How? Where do we go from here? This webinar will take place on Tuesday, June 12th at 2 p.m. Eastern Time. Once again, thank you for joining us and enjoy the rest of your day. Take care.
Thank you.